If you're shifting to the right, you're unbinding oxygen from your hemoglobin molecule. If you unbind, that means you have too much CO2. Perhaps you're in acidosis as well. So your body's going to shift it where you actually unload more oxygen to the body cells and tissues, because the assumption here is that you're using that oxygen to turn it into ATP. So unbind oxygen when you have acid conditions, excess levels of CO2, or warm. Other questions?
When you talk about the trace part of the assay, is that for 1 on page 7?
Let me see. Yes, it is part of that on the bottom of 77. What you want to do is take the air in, take the air out, but also think about how it leaks to the whole heart blood flow that we did back in chapter, what was that, 18? So it's not just that, it's actually add to it the blood flow.
But it's already on D2L?
It is on D2L, yeah. Did you have another question?
I'm not able to open up those pages on D2L.
You can send me an item. Actually, CNET is a website, can actually help you download some stuff to open those up.
Yeah, it's called IRFAN, IRFAN. If you just download that program, you can open them all.
It's an old type of flash animation. They typically don't use those anymore, so yeah, use the IRFAN to open them up. I don't know why they don't support those much, but...
Okay, do you want to know both of those at the bottom, or just the...
I would know both of them just to be on the safe side, because one class gets one class, the other class gets the other one. So depending on which town you take the test, you might get one or the other.
So we need our fan to open it?
If you open those animations, like the ones that I'm showing you, yeah.
Page 77.
How about other questions?
And where do you work? I couldn't find the pathways yet.
Did y'all do that in lab? No. Basically, if you follow through the course path, it's going to give you that pathway, more or less. Why don't you try filling it in, and then let me check and see what you've got. Okay. And I'll be happy to do that for you after class if you want. Let me get you started back in the course pack on page 95 with the digestive system. We've got 2 major organs to finish, and then a few of the digestive accessory organs to look at. I'm on page 773 in your text. Well, in class on Tuesday, we talked about the stomach and its activities. The stomach was responsible for chemically digesting what types of food groups? Mainly proteins. Yeah, mainly proteins. How did we digest proteins in the stomach? Pepsin, good. We also could use rennin. Rennin breaks down a specific protein known as milk protein. Good. Rennin breaks down milk protein. What was the pH of the secretions from the stomach? 1 or 2. Yeah, 1 or 2. Really, really acidic in terms of pH scale. Well, as the food is channeled here into the small intestine, food can spend anywhere from around 3 to 6 hours moving through the 20 or so feet of small intestine tubing. We call it the small intestine because it is in fact smaller in diameter than the large intestine. And you see in this picture we have both small and large intestine kind of grouped together. You'll notice that coming up across the top here is the large intestine, kind of wraps around the outside of the small intestine. Where your belly button sits is approximately where transverse colon and jejunum are found. So if you have an ache around your belly button, it's not actually your stomach that hurts, it's actually portions of your small intestine that may be giving you some problems. So, notice here in your worksheet, the three major subdivisions are the duodenum, sometimes called the duodenum. That duodenum is the main place where digestion and absorption occurs. Since it's the main place for digestion and absorption, we have the most modifications to enhance digestion and absorption in that duodenum. What portion of your small intestine is duodenum? How much?
10 inches.
Yes, 10 inches. We're not even talking a foot, right? We're talking a very short piece of your small intestine is that first duodenum. The next region is called jejunum. That's the middle region of your small intestine. You'll notice that's approximately 8 feet. And still, we're going to have villi for absorption. We're still going to be doing digestion there. But as we end the near of that jejunum, near the end of that jejunum, we're going to run into the ileum. The ileum is going to have the least amount of modifications for absorption and digestion. Keep in mind, ileum sounds like the ileum of your hip bones. That kind of is not a mistake. So it, of course, is the lowest or the last portion of your small intestine that's going to hook in with your large intestine. There's two sphincters that regulate passage of food through your small intestine. What is the name of the sphincter that separates stomach from small intestine? Pyloric, good. And then, of course, the one that connects your small and large intestines is what?
Ileocecal.
Ileocecal. They sometimes call it the ileocecal valve. It's still a sphincter, but its job is to regulate the passage of food from small to large intestine. Now notice in total we have around 20 feet of small intestine. That's been measured in a cadaver. Dead people have no muscle tone. So in living people we do have muscle tone, and our small intestine really truly isn't 20 feet. The muscles stay at a very mild state of contraction, and so in a living person it's really less than 20 feet. Let's take a look on the inside of the small intestine. This looks somewhat familiar. Inside of your stomach, we talked about simple columnar epithelial tissue. We still have that, but this time it's convoluted into these finger-like extensions known as what? Villi, good. Which organs have villi? The digestive organs, that's right. That's a smart ass answer, isn't it? I should say which specifically have the villi. There you go. Intestines have the villi. Large intestine actually has some. Small intestine is the major site for those villi. Stomach has none. So stomach has convolutions known as what?
Rugae.
Rugae, good. So we've got the villi in the small and the large intestine. The whole point here is to help increase surface area. If you have 20 feet of small, roughly 5 feet of large intestine, then what we've got is 25 feet of tubing. But that's not enough. We actually need more convolutions to increase the internal surface area. So imagine if you were to take this and then stretch it out flat. Imagine how much more mileage you get just by having these internal convolutions known as villi. So we greatly increase surface area with these finger-like extensions known as villi. That way we can maximize the amount of absorption and digestion that occurs here. Your small intestine really is the main organ for breaking down all the food groups. It's also the main organ for completing the absorption process. So this is where the activity occurs. That's why we have to have these modifications, not just for absorption, but also to assist with digestion as well. If you look closer, you will notice there's teeny tiny little projections on each individual villus. These are called microvilli. Do you see some of them kind of pooch out there? These are the microvilli, and they further help to assist with absorption and digestion. So those two activities are key for the small intestine. Large intestine has some villi, but the large intestine has absolutely no microvilli. So we're going to find the microvilli are special for your small intestine. Some people say this looks like a brush border. You ever look at your tongue in the mirror? Looks bumpy, looks like it has hills and valleys. That's kind of what they mean by brush border. The small intestine surface has that same bumpy appearance. So this actual surface here is a great hiding place for enzymes and digestive juices to wiggle their way down between these villi and continue the process of digestion and absorption. One more thing that we've got here, we've got circular folds. Notice we call them plicae circulaire. That's a fancy name for them. These are nothing more than speed bumps. They're going to slow down the process of food being moved through your small intestine. So it's not the villi or the microvilli that create these circular folds.